Let me start while I load up my slides by thanking the organizers and everyone for attending today. It's very kind of you to give me the opportunity to review some genetics 101, if you will. And I also want to thank Emma for giving a wonderful start to the conference. And as Sangeeta mentioned in her introduction, this is not a perspective that we typically hear in most of the conferences that we attend. And I think that it really resonated with a lot of people. And it's an important consideration that we should keep in mind as we go forward. Let's see, let me kick over to slideshow. And I think everybody can probably see the slides presently. I've been asked to discuss what is CDKL5. It's a 3-word question. It would seem very simple, but in fact,
sorry, James, I don't see your slides.
You don't?
No.
That is unfortunate. Let me see which button I did.
Maybe just click share and then try resharing.
I did. Okay. How
Okay,
about now?
I see them now. Yes.
Okay.
Just go to. I see your edit screen. If you just go to full presentation.
The first hiccup of the morning.
That's perfect. Thank
you. Okay, now can we see them? Looks good. Okay, so let me go back to that simple 3-word question of what is CDKL5. And in fact, it's a large question, and it's a question that's of great importance for anyone who has an interest in CDKL5 deficiency disorder. And I thought probably the best way to highlight the importance of this is to share a quote from my friend and colleague, Professor Joe Zhao from the University of Pennsylvania, who is one of the leaders in the CDD field. And Joe has stated that understanding what CDKL5 is and does, as well as how, when, and where it does its work, are key questions that remain largely unanswered. Figuring out these aspects of CDKL5 is needed to guide the development of future treatments. And I really think that hones in on this whole concept that what is CDKL5 is anything but a simple question, and it's well beyond the scope of what one could cover adequately in a single presentation today. So what I'd like to do is to focus today on 2 aspects of that larger question, which are the genetics of CDD and the function or functions that we know are attributed to the CDKL5 protein. So the first thing I'd like to do is to actually distinguish between what is the CDKL5 gene and what is the CDKL5 protein. So as I'm sure you're all very well aware, a gene is a sequence of your DNA that's present within the nucleus of your cells, every cell in your body, or almost every cell in your body. And the CDKL5 gene is actually present on the X chromosome, which is the chromosome that defines your biological sex. Now, the DNA itself is comprised of 4 different bases, and the order of those bases, or the genetic sequence, is what provides the blueprint, if you will, or the instructional code that eventually leads to the production of the CDKL5 protein. If we then move to the protein itself, proteins are just simply assemblies of different amino acids, and those are the building blocks of the proteins, and they're joined together in a tandem array. And there's 20 different amino acids that can be utilized for forming proteins, and the genetic code is what defines which of those amino acids will be placed in which order. And once assembled, the protein is what actually does the functional work. So the gene itself doesn't do work. The gene provides the blueprint for what eventually gets synthesized in the cell that then does the work. Now, the DNA code itself, as I mentioned, is what defines the amino acids that will make the eventual protein. And the DNA code itself is the so-called triplet code, whereby a tandem of 3 bases is specific in its sequence for an amino acid. So in this cartoon that's illustrated here, the GGT sequence will specify an alanine amino acid. The CCC will encode for a proline. TGT will encode for a cysteine, and so forth and so on. the proteins that are synthesized are these linear arrays that can be either long or short. Short proteins are often referred to as peptides, and functional peptides can be as short as even 2 or 3 amino acids put together. But other proteins can be quite long. CDKL5 protein itself is just a bit over 1,000 amino acids, and it's on the larger end of what is typically seen for proteins in the body. It's certainly not the largest, but it would be a little bit above what would be the average size. So it's a lot of amino acids put together, and the code that provides the blueprint for the order of those amino acids constitutes the CDKL5 gene, which resides in the nucleus. Now, I like to use the analogy of proteins and trains being quite similar. I think it's a good analogy whereby the train itself is comprised of different cars, and the cars themselves would be analogous to the amino acids. And the types of cars that are present in the train, and to some extent, the number of cars, will actually define what the function of that train will be. The design of the cars comes from an engineer or a director or some company executive. Well, that would be the same as the gene itself in the nucleus. And as I'm sure everyone is well aware, that the cars that are present in a train are put there by purpose, and it's for the needs that the train will be using to deliver its cargoes. Now, the genetic code, as I mentioned, is what defines the amino acid sequences. And so that's illustrated here, where I put blue bars over the triplet sequences. And so in this case, this is actually on the reverse strand of the DNA, but the code illustrated here would be an AUG. Well, that always encodes for a methionine amino acid. And just like all trains start with an engine, all proteins start with a methionine. But then downstream for that, the triplet code defines the different amino acids. And I've just illustrated a couple of different types of cars that could be put in an array to form the body of the train. Now, sometimes things happen, and that DNA code can change. In this case, I've illustrated the C base that's on the first position of what would be the normal triplet code, the CCG, which would normally encode for a glycine amino acid. But sometimes, as I mentioned, things happen, and that can change the amino acid that would be encoded at that position in the genetic code or the blueprint. And on this side, what's illustrated is where a base is changed. So the C gets changed to a T. And that T changes the glycine amino acid code into a serine amino acid code. So that would be a G to S change. Now, it could be a mutation. Mutations mean that there's a consequence. The function of the eventually encoded protein would be altered. But not all base substitutions are mutations. Some are just polymorphisms. You change the code, but it doesn't affect the function of the protein. And we have all of these. There is a human genome project, but there is not a single human genome sequence. All of us are different. All of us have many different changes in the base sequence throughout our genome. It's only those that have some consequence and a deleterious consequence that would be labeled as mutations. Another possibility that could happen is that a base is lost. And in this case, the third base, the A, that's in the preceding triplet codon, which would encode for a phenylalanine, is lost. It's deleted. So this would be a one base deletion, in which case you see the triplet code changes. It's the same for the first two amino acids in this illustration, the methionine and the lysine, but now instead of being a AAA codon sequence, it's now AAC, because the A is lost. The C that would be the first base of the second triplet codon now becomes the third base, and every amino acid downstream of that is changed, because that would be a frame shift mutation. And obviously, an incorrect positioning of amino acids can affect the functionality of the protein, and that can be deleterious to the function of the system. And if we go back to our train analogy, What I've highlighted here is a passenger system, if you will, if you're from an area that has a subway system, a subway car, where if you change one of the passenger cars into a rail car, you're not going to transport as many people. You're going to put a hold on the system. People are going to start backing up on the subway station, and that is going to have consequences. And it may very well affect the function of the entire area around the subway because people that should be getting to work on time aren't. And so that's another way of thinking of how these mutations function. At the most extreme end, you would halt the system completely. But it doesn't have to be a complete shutdown of the system. It can be a hindrance, or we call that a hypomorphic effect. It works less well, and it still has consequences. And this is important in the context of CDD, because we know that there are many different mutations in the CDKL5 gene that do alter the amino acids in the encoded CDKL5 protein. And illustrated here from a study that was published several years ago from Ralph Hector are the identification of different types of mutations of CDKL5 and where they occur in the gene. So the top part here is the genetic structure of the CDKL5 gene on the X chromosome. It is a complicated gene. It has a complicated gene structure, but you can see that there's different types of mutations that are causal for CDD, and those are illustrated in red and probably yellow, that don't necessarily occur at the same place, but they all have a pathogenic consequence. Okay, so that then leads us into what is the function of CDKL5 protein that gets interrupted by these different mutations, and then what physiologically does CDKL5 normally regulate. And so we move now into the area of our initial what is CDKL5 question, where we probably know less about CDKL5 than many other proteins that have been studied in different contexts. We do know that CDKL5 is a protein kinase, and I'll talk more about what that is in just a moment, but we have to recognize that there may very well be other functions that remain to be discovered. Physiologically, we now know that CDKL5 stabilizes the cytoskeletal structure of cells, and I'll talk more about each of the first three of these in a moment. But that's not the only thing that we now recognize CDKL5 does. We also know that CDKL5 can help with DNA repair, particularly by stabilizing segments of DNA that are active when they need to be repaired. And we also now recognize that CDKL5 helps regulate the action of cilia, which are hair-like structures that help move the fluids outside of the cells and outside of the tissues throughout the body. And recently, there was actually a study that came out that suggests CDKL5, when overactive or in certain contexts associated with disease, may actually contribute to the pathology. So I won't be talking about the pathogenic role of normal or of functional CDKL5 function of activity today. But there's also other contexts in which too much CDKL5 action may actually be problematic to itself. And as I've said a couple of times already, we still have to recognize that there may very well be other things that CDKL5 does that remains to be identified. Now, with respect to the protein activity of CDKL5, yes, we know that it's a member of the kinase family. There's lots of kinases in your body, hundreds, if not thousands of different kinases, and they all have a similar amino acid sequence that forms the catalytic unit that's responsible for doing the kinase work. And on the CDKL5 protein, that domain is located towards the starting point or the amino terminal region of the encoded protein. So as a reference to our train, this first base would be the engine. It's the methionine in CDKL5. And then just shortly downstream from that is the kinase domain. And we know quite a bit about the amino acid sequence there and which amino acids are critical for the preservation of the kinase function. Downstream of that, where mutations that cause CDD still occur, we know a bit less about what that region of the protein does. And there's a lot of interest currently in trying to delineate what the different roles of the outside of the kinase domain regions of the CDKL5 protein do functionally. But getting back to the kinase itself, the kinase takes an energy molecule, ATP, and transfers its phosphate onto a substrate. Illustrated here is a protein. The kinase then places a phosphate group on an amino acid of that target protein. And as I mentioned, there's lots of different kinases. Some kinases recognize many proteins. They're very promiscuous and they will phosphorylate many things. Other kinases are very specific in their actions and require a very highly selective context to promote the phosphorylation of the target. This is one of the areas where a lot of effort is currently focused, is to identify what the targets of CDKL5 are and what those targets do when either phosphorylated or when non-phosphorylated in different biological settings. And by no means is this a complete chapter of the book. In fact, a lot of investigations are required to fully delineate the total spectrum of targets that are recognized by CDKL5. But there have been several. Sorry, before I get to that, I just want to highlight the importance of the kinase domain in the pathogenesis of CDD, as the majority of the different mutations that are recognized to cause CDD do affect or do reside within the catalytic region of the CDKL5 protein. So each of these highlighted in red are known mutations that do affect the catalytic activity of that kinase motif. They're not all 100% abrogating a function. Some of them will change how well the kinase works, but it can be the full spectrum. It can be a complete abolition of kinase activity or a hindrance of kinase activity, either of which, as illustrated in these red different mutations, are sufficient to cause the CDD phenotype in patients. Now, getting back to the targets of CDKL5, as I mentioned, we have recognized that CDKL5 modulates the cytoskeleton. And so in this cartoon is a schematic of a neuron, and the cytoskeleton is like the framing of your house. It's on the outside, it's on the inside of the cell, but it supports the membrane and the things that are trafficked up and down the cell. And illustrated in red are the modifications to that cytoskeleton system that can be conveyed by CDKL5, which is phosphorylation. And known targets of CDKL5 that relate to cytoskeleton function are shown here. So MAP1S, EB2, and CDKL5 itself are reproduced by different labs and are consensus recognized targets of CDKL5. And yes, CDKL5 does phosphorylate itself, and its autophosphorylation does change its catalytic activity. Okay, so that's the cytoskeleton. But that's not the only targets of CDKL5 that have been thus far identified. In fact, there have been several targets that are found in the nucleus of the cell. And this is where the genetic material is. This is where your DNA lives. And so it would suggest that CDKL5 also plays a role in regulating either gene organization, nuclear structure, or even perhaps how genes are active or inactive at specific times in the cell. So there's different proteins that have been reported to be phosphorylated by CDKL5. As I mentioned, those three in bold have been verified by independent laboratories, but we have to recognize that others likely exist and remain to be identified, and their roles fully examined, both with and without CDKL5 phosphorylation. Physiologically, if we again look at our schematic of a neuron, which shows the terminals, the endpoints, this would be where the synaptic or the communication between cells takes place. One neuron would give a signal from this end, and it would connect with the dendrites of the other or recipient neuron. And if we magnify this, what CDKL5 does is it allows for these microtubules to grow, to mature, and to form a functional communicative system between this process and what's not shown here would be the process from the other neuron that is connecting to allow an efficient communication system in the brain. In the absence of CDKL5, it's not that this doesn't happen, it just doesn't happen as well. The processes still form, but they don't fully mature, and their communication is inefficient and perhaps unstable. And so that by sort of by extrapolation suggests that CDKL5 is critical for this normal maturation, development, and synaptic establishment process to properly take place. But as I mentioned, there's other things that are now being identified as being targets for CDKL5 and regulated by CDKL5. And a couple of the more, from my perspective, fascinating aspects, one of which is this regulation of the mobility of cilia. As I mentioned, cilia, shown here in an electron micrograph, are just like they look. They're hair-like structures that vibrate or move, and in doing so, will move the fluids that are present on the outside of the cell. And often they do it in a specific direction. So they're a rhythmic type of beating, if you will. Now, what that does is it removes the bad material that's been secreted by the cells and pushes it down for excretion. And it also brings back good stuff to nourish the cells. Without CDKL5, the beating of those cilia is errant. It's not as efficient, it's not as rhythmic, and so the fluid doesn't get moved in the proper directions. And there has been some suggestions that CDKL5 deficiency disorder may actually be a member of the ciliopathies, which are conditions which are recognized specifically to be caused by the immobility of the cilia. So in addition to playing a role in the maturation process of neurons themselves, it's now recognized that CDKL5 protein also contributes to this other function where impairments are clearly recognized to be associated with a pathogenic sequence. And finally, John Rouse's group in Scotland has recently identified that CDKL5 may also participate in a DNA damage response system in cells. As I'm sure everyone's aware, every day we're bombarded with cosmic radiations and other environmental and natural events that damage our DNA. And the system that we have is a wonderful system because it repairs that DNA damage. And these are some of the participants in the repair systems that are present in every cell of our body. What John's group found is that CDKL5 phosphorylates these factors within the nucleus, and those are the ones recruited to the site where that DNA damage repair is taking place. Now, what it seems to do is it silences those regions. It sedates them, if you will. It's like the anesthetist that puts the system to sleep so the surgeon can repair the damage. You wouldn't want your DNA to be too active when the repair systems are in progress. So what CDKL5 does is it seems to calm the system and allow an efficient repair to take place. In the absence of CDKL5, it's not that the repair doesn't happen, it just doesn't happen as rapidly or as effectively, and it takes more energy and time. And so CDKL5 now seems to have a role not only in the maturation process of neurons, in regulating the activity of the cilia, but now also potentially in how well and effective our DNA repair systems work. And so to summarize, I'd like to go back and just highlight that the DNA sequence of our gene is what contains the blueprint for making the eventual protein. The code specifies which amino acids will go in which position of the eventual protein, and mutations that cause CDD occur within this gene sequence. So all of the mutations actually will affect the DNA. Those mutations then change the encoded blueprint and will allow an improper amino acid sequence of the CDKL5 protein to be formed. Now, the protein itself is normally a kinase that has a number of targets and regulates different physiological processes. It's critical for brain development and communication. We recognize it plays a role in the normal function of cilia, and we now recognize that it is involved in DNA damage repair. As I've mentioned before, though, there may well be other functions of CDKL5 that remain to be identified. And as Joe Zhao mentioned at my first slide, those identifications will be key to developing future therapies that will more effectively manage, if not cure, the CDD itself. Finding the most effective treatments for CDD obviously requires a full understanding of everything that the CDKL5 protein does and what goes wrong in its absence. I can say 15 years ago, we knew embarrassingly little about what CDKL5 does. We know much more now. There's still a lot to be learned, and great progress is being made in all of these areas. And I'd like to end by just acknowledging three organizations that have helped our work in Toronto: the CDKL5 Canada, the IFCR, and the Lulu Foundation, who by providing funding and support are allowing some of these advances to be made. And with that, I will stop, and I thank you much for your attention. Hopefully I've been able to shed some insight into the genetics and the role of CDKL5, and I'll be happy to try to answer questions.